Good afternoon and welcome once more on behalf of the Center for Clinical and Translational Sciences and within the center. I hope all of you are members. If not, please do consider signing up. Lots of benefits to go around, so do take the time to sign up if you're not a member already. But within the center, we have one of the key emphasis area is to find and support and develop new technologies that would be effective in driving translational and clinical research. So there is a program on that, and I direct that program together with the program on pilot studies. And this session is primarily to showcase some of the strengths we have at Ohio State at this time, and for you to tell us some of the strengths you have. So the format would be, as you know, the previous session ran a little bit late, so we are starting a little bit late. The format of this session is where each speaker would speak for about 10 to 12 minutes, no more, so they tell you the big highlights of their program. And then we save ourselves at least about 20 minutes or so where the speakers would come up, and then there would be a discussion session. Within this discussion session, of course, you can ask the speakers about what they spoke, but you could also bring up matters that are relevant, technologies that you are doing and you would like to see be supported. And this is a forum for bridge building and networking. So if you would like to work with our speakers, take the opportunity to meet them, figure out how you could use their cores if they have one, so on and so forth. So this is supposed to be a very interactive session. This is not one of those lecture sessions where you just listen, take notes, and at the end. So about 10 minutes for each speaker, and then after that, the speakers come up here, and then we have an interactive discussion. With that background, today we have 4 speakers. We have Dr. Raghu Machiraju, and Dr. Machiraju is a professor at the Department of Computer Science and Engineering. He's from the College of Engineering. Then we have Dr. Kun Huang. Dr. Huang is an associate professor in biomedical informatics. Then we have Dr. Pearlie Yan, who runs the core, as you will hear from her, and she's from the James Cancer Center. And finally, from the College of Arts and Sciences, Department of Physics and Biochemistry, we have Dr. Ralph Bundschuh, and these will be our 4 speakers. They will talk about their technology platforms in general, and they will avoid getting into minor details, and that part of detailed discussion will be kept for the discussion panel, which will happen just after the speakers are done talking. With that, I would like to invite Dr. Raghu Machiraju to talk about tools for quantitative phenotyping. Raghu.
Thank you, Chandan. Let me give you a brief introduction to some of the work we've been doing. So I'm really, a computing type. I don't know much biology. I do want to make the disclaimer. And develop a lot of software, did contribute to NIH, NLM-funded software. Now, let me say something about the kind of phenotypical analysis we are looking at. As you all know, there are many different kinds. We've been mostly focusing on morphology. Sometimes we did kind of veer into functional phenotypes. Like we've done quite a bit of work on functional MRI, data analysis. We're starting to do more and more white matter stuff, along with Cameron and so on. Now, but today I'll be mostly talking about the cellular phenotypes. And for example, if you take a dynamic in-vivo system, like the wound, you can see the panel here. And of course, if you take tissue samples, you see a lot going on on different days, and depending on the condition, non-ischemic or ischemic, there's a lot of, remodeling at the tissue level, and maybe even changes at the cell and nuclear level. So how do you even try to get some handle on this? So, of course, we start with let's study angiogenesis, and maybe through those mechanisms and by looking at certain cellular behavior, maybe we can get some handle. So we grew some cultures, and once again, I'm a computer scientist. My students don't know computer science, but we got into all this stuff, and now my students know how to grow cell culture. So it's been a wonderful journey, and we're learning new things. And we also, in addition to learning new biology, for us at least, we started using some of our techniques which we have developed for many, many years. Could we actually, perhaps look at things differently, not just 2D slices, but or 2D slides, but actually extract 3D structures. Like, for example, we started extracting the 3D structure of the nuclei. How do the different nuclei of endothelial cells, how do they look? And we use confocal microscopes to get these images, and we of course noticed that there are 3 different types in these endothelial cell cultures. And then if you start doing some simple analysis at the 0th hour, you see a certain distribution of curved, round, and flat. But after the passage of 8 hours, you see that distributions have changed. So why, how? I mean, you can ask all those questions. Can you generate hypotheses? Can you look at these phenotypes and perhaps think about developing some platforms, right? So some of the funding we got from CCTA was actually used towards this purpose. And so instead of just kind of eyeballing and using manual methods, we started doing actual data analysis, like fitting simple things like ellipses to these cell shapes, and then start using some statistical methods like multidimensional scaling, blah blah blah. I mean, and then start seeing if there are any patterns. Turns out there are patterns. So we started, for example, if you look at the red dots and the blue dots, they're 2 different timestamps. And yes, I mean, some of these shapes, the diameters, okay, of the ellipses, they form certain patterns. So we went further and said, why not extract complete 3-dimensional shapes of this nuclei, and then maybe do some real, consistent statistical, analysis. Can we say something about the 2 different, timestamps? Can we talk about the distributions? And you can, and we are starting to see differences. And a lot of these kind of analysis really lead actually play into something called single-cell analysis. That's a big topic. It's one of those cross-cutting themes in NIH now. And the essential argument is that let's not worry about averages anymore. Let's look at multivariate analysis. Let's actually try to look for subpopulations, and that's probably the way to go instead of just trying to get some a big clump of tissue and then doing some kind of global analysis. So we are playing well into this bigger trend that's coming out of NIH, but a lot of our work started in the tumor microenvironment. We've been working with Gustavo Leone and Michael Ostrowski for many, many years, and we started our exploration of the fibroblasts, macrophages, endothelials in the tumor microenvironment, and they were essentially trying to understand what happens when you knock off P10 and P53 in the breast tissue, okay, of mice. So, I mean, this was a very comprehensive study. We looked at different cell types, there's a lot of genetics involved, and so on and so forth. And of course, we used transgenic mice to actually extract all these cells and then analyze them. And we analyzed them using a pretty comprehensive workflow. I mean, essentially, this is like a 3D workflow where we, locate the cell through segmentation methods, and this come out of confocal microscopes, and then we do all kinds of segmentation, and then actually extract a 3D shape, and then compute many features, measurements on the shapes, and create a gallery and then do group analysis and do all the, the stuff we can do, but with huge populations now. Okay, we can do principal component analysis of the shapes and so on and so forth, and you could talk about how the volume, elongation, flatness, all those different properties. You could try to start describing the nuclei. You could do the same for the entire cells too. I mean, so this is just a way to demonstrate that many of our tools can be used in biology, that computer science can play a role, and that's the essential point I'm trying to make here. Then, of course, you can start classifying, shapes, using these descriptors, and then not only just shapes. I mean, you could bring in, the actual chromatin content, and you can use, in addition to the chromatin content, of course, you can use the shape and the context too, where these different nuclei are present. Then you can do all kinds of subpopulation identification. For example, in our tissue samples, we found that there are 3 different classes of fibroblasts, 3 different classes of macrophages, and 2 different classes of endothelials. And you could do that using our kind of techniques. And not only that, you could even talk about how the phenotypes change. I mean, you could model it as a graph, and these are the things we know really well. So how do small elongated nuclei, these P4s, how do they become flat and large? I mean, what is the percentage of these that become flat and large? I mean, all these kind of questions you can ask. Okay, and not only just at the nuclear level, we could do the same with the tissue. I mean, this is on the project we are working with somebody at Penn State, where he basically does direct genetics. Okay, and using different assays, he creates all these phenotypes, and some of these phenotypes have of the zebrafish. Some of them have big eyes, some of them have small eyes, and essentially the goal is to look at all these phenotypes together. Okay, can you actually organize all these phenotypes? Here you have an organization of 160 phenotypes, and we're looking at the eyes. So we did some feature extraction, we did all kinds of analysis, and then we used a certain tree, and trees are something we know really well in computer science. So all the ones which are close enough, they all cluster together. The ones which are not close enough, they are further away from each other. And another thing that we could also do with something like this is to actually, look at the labeling of pathologists. So what you see here as different colors, red, blue, green, these were the labels that a pathologist, ascribed to the different, samples. Right? So we could see that, you could see how good the pathologist was or how bad our feature estimation was. I mean, there are many, many things like that. So these kind of trees can really help with large-scale phenotypical analysis. And essentially, we are doing what's called dictionaries. We are looking for words in a dictionary in the phenotypes, and we try to use some statistical measurements to create those trees. And you could not only do it for disorganization or organization at the tissue level, but you could look at hypertrophy, you could do necrosis, all kinds of stuff. And this is basically what we did. And of course, we had to build many, many tools. And that's what we're really toolsmiths. And of course, databases, and almost like a limb system we had to build. And now we are trying to relate it to the biology. Can you relate shape to gene expression changes? Okay, can you actually use some of the ideas we learned in nuclear shape morphology, what happens in angiogenesis? Can you actually relate that to what's going on in the nuclear envelope? I mean, we're trying to go back to the biology as much as possible. Then can you apply the same kind of ideas, not to 3D stacks anymore, but could you actually do it on H&E images? I mean, so on and so forth. And again, even looking at different modalities, not just confocal microscopes, can you actually do it on micro-CT? So that's basically what we've been doing. Many, many tools, many kinds of images, and not just images. I mean, I work very closely with Kun, so we also do the similar kind of analysis on molecular expression. So that's pretty much what we've been doing for the last few years.
So, thank you, Raghu. So if I understood right, Raghu is trying to tell us you are what you look like. So if you have projects related to phenotypes, then this group has the technology for objectively tracking it and giving you traction. It's easy to say something is big, something is small, something is flat, something is long, but to objectively and comprehensively capture that is not trivial. And this group specializes in that, so I would encourage anybody that deals with, as you saw, it is not just at the cellular level, but organismal level, and I'm sure it will be at multiple levels, be it biological, be it biomaterial. This is a go-to group.